morning, everyone. My name is Luiz. I'll talk briefly about what is HERV-W, endogenous retrovirus, and multiple sclerosis, and the link between these two factors. So, HERV is the abbreviation for human endogenous retrovirus, which has a simpler genome compared to nowadays exogenous retrovirus such as HIV. However, it has all the classical genes, GAG, O, and ENV. HERV-W is member of the gamma retrovirus family, which also has the murine leukemia virus and feline leukemia virus. So, endogenous retrovirus, exogenous retrovirus, let's understand a little about it. So, sometime of the history, an exogenous retrovirus infected our ancestors' germline cells, and it passed through the generations until now. And I expect, specifically, HERV-W infected our ancestor germline cells around between the age of New World monkeys and old World monkeys. So, now we know that at least 8% of our genome are composed by endogenous retrovirus sequences. However, most of them are silenced because they are most truncated and does usually express in our genome. However, again, sometimes it can be expressed. It also has some physiological activity, such as the protein syncytin, which is a very important protein during the pregnancy. So it also can be expressed and it can be linked with some diseases, such as cancer and autoimmune disease, and it more specifically and more important for us, multiple sclerosis. So, multiple sclerosis is an autoimmune disease with no etiology. However, it's believed that the virus is the main candidate for its etiology. We have some viruses candidate here, most of Epstein-Barr virus and HERV. It also, the disease has an inflammatory stage, which is very similar to other viral infections, such as HERV. The disease is also characterized by its progressive and degenerative course of disease. We also know that more than 1.3 million people has multiple sclerosis in the world. It's more frequent among the women and the white people. However, the course of disease is much worse in men. So, now talking about these two items linked, what do we know until now? We know that HERV-W is highly expressed in MS patients. In all these materials, blood, brain, and CFF. Our group and other groups also reported that higher mRNA levels in MS patients when compared to others healthy individuals. One important thing one important finding that we very recent, that natalizumab is one of the best drugs for multiple sclerosis treatment available today, and it was able to reduce significantly the expression of HERV-W in MS patients, reinforcing that could be some link between HERV-W and MS. Also, a collaborator of ours, Home, also reported that ENV protein is highly immunogenic. It was able to induce an important and high inflammatory response in vitro. Our group reported that HERV-W ENV protein and MOG, myelin oligodendrocyte glycoprotein, are similar. We could see that at least 5 sites of similarity, which could suggest that maybe autoimmunity in this disease could be caused by molecular mimicry way. As I told before, we could see high levels of mRNA expression. However, it was able to detect the envy protein in demyelinating lesions in MS patients. And then one of the most important findings that is still in press, I guess, so that mice that were exposed to HERV-W envy protein come to develop experimental allergic encephalitis, which is the animal model for MS. So we have many findings here that comes to the question: is HERV-W only a coincidence for MS? Yes. Oh no, maybe not. We believe yes. So what do you know until now? What we must to consider to further studies? The autoimmune process in MS is very complex. It's not fully understood. Also, we don't know how is the profile of HERV-W expression in MS. Which transcripts are really expressed? How is the frequency? Some transcripts express more than others. Do we know all the transcripts in these patients? Also, what is the really role of HERV-W in MS? On the other hand, we know that we don't know how the immune system works essentially in the MS pathogens. Also, which cytokines, pro-inflammatory, anti-inflammatory cytokines are more expressed than others? How is this inflammatory response essentially? How is it? There are other immunological genes also. So we have so many questions to answer. In consider all these questions, I think we need to study the disease more globally. And that here comes the idea for my PhD project. Which unfortunately, as Franz said, we still don't have any results. So, which is entitled "The Expression Profile of HERV-W in MS Patients Under Relapsing Conditions," under the disease in its course itself. So, which is the aim of my study? Mainly to perform the transcriptome. Well, we'll see the transcriptome for HERV-W, other HERVs. We can see it will be available for us, and also for anti-inflammatory and pro-inflammatory cytokine genes. Mainly to see the disease essentially in this point. And also we will compare the data between MS patients under these conditions and the results founded on healthy controls. For that, you have two groups. The control group, which will be composed with healthy patients without any familiar history of MS, and MS group, which will be composed of MS patients under relapsing conditions. For that, we will collect 10 PB cell samples of each group and the samples will be submitted to mRNA extraction. For that, for the transcriptome itself, we will perform the next generation sequencing, which will be through ion proton platform. And just to have an idea, we can have more than 10 gigabytes of information. This will be ideal for whole transcriptome analysis, not only for our focus on this study, but we hope that this data will provide the research for at least 5 or 10 years. For the transcriptome analysis, the assembly quantification and SNP variants, we work with these programs, CLC Workbench, Genius, and Mira. And for mapping the sequences into the chromosome of our genome, we will be using BLAST2. So, we're still not very familiar with this transcriptome analysis. But first of all, we start to work with these programs, maybe in the future have more options and for specific analysis. Well, I would like to end my talk thanking especially for Virology Lab from here. My coordinator will be, who must be mad by now because she said, don't dare to put a picture of me in your presentation. And the Camila's team, which is composed by Paulo Urbano, Felipe Scassi, Ana Carolina Soares, Paulo Nasser, Cristina Nuni, Franciele, Deborah Gerhardt, and Giovanna Calais. Thank you very much. Any suggestions or questions? I'm here.
Question, in that natalizumab decreasing the HERV, was that in the CSF, cerebrospinal fluid, liquido, or in the blood?
Blood.
So they didn't look in the...
I'm not really sure, but the most impressive finding was really in blood.
Interesting. Because it's thought to work by decreasing T cell trafficking into the brain, right? I mean, that's what natalizumab does. So how is it decreasing the HERV expression then?
Well, essentially they didn't explain it indeed why it happened. It's believed that the HERV-W can be the source of etiology, and they looked for the natalizumab, which it's a really impressive drug nowadays that can even decrease the oligoclonal band that is usually found in MS patients. And they thought, we also did, but they were faster. And they also thought about that and tried to look at HERV, and they really found that it decreased. I don't have it here, but I can send you the article later.
Actually, natalizumab is, nobody knows why exactly, because the main function is to block the migration of T cells to the brain. But natalizumab is also able to change the expression of some genes responsible for differentiation of the cells. So we believe that not only these genes are affected, but also HERV-W or other genes that may be responsible to transactivate HERVs, or we actually don't know. But natalizumab do not only block
That's another question.
Sure.
So, this betrays my ignorance, but why HERV-W and not HERV-K or other lineup? Why is it that particular HERV?
Okay, so I don't know exactly the answer for that. What we know that when they start searching for some virus that could be the candidate, they do not find they didn't find any expression or HERV-K or other HERVs. Essentially, HERV-W.
Yeah.
Is that right?
Is there any way to work in this model looking at impact of the computing on T-cell activation?
Actually, they did. I'm really sorry, I'm not an expert in immunological experiment. But as far as I know, Perron tried to, in vitro, they see, a group C saw that HERV could activate 2-octa receptor 4, essentially. And many others that are, I'm really sorry, but I really don't know how it is.
But you know much better than I, because
okay.
anything about it.
So, as far as I know, they have some experimental models for HEMS protein. But they really can activate 2-octa receptor 4 and more inflammatory responses, but that's only what I know.
So it's not just that in those MS patients there's whatever mutation or deletion or whatever change to
promoter, or actually the promoter of HERV-W and whatever molecule might be downstream of that. It's really an effect of the envelope protein or the virus itself that triggers T cell.
Well, I believe it's the envy, really, essentially. But because it's the more exposed, then in more immunogenic between the older portraits. But we will look for everything there. Maybe we can find something new here.
Muito bem.